My name's Amber Reinick,
I'm Sabrina Elliott.
and our enzyme is Kallikrein 6.
So first we're going to address the familial information of Kallikrein 6. It belongs to what's called the tissue kallikrein family. There are 15 structurally homologous enzymes within the family. This is the largest contiguous grouping of serine proteases in the entire human genome. They're all located on chromosome 19 on the small arm in section 1, band 3, subband 4, and they're located throughout the body, participating in a diverse functions, and they all participate in proteolytic cascades with trypsin or chymotrypsin-like activity, and they're infected by endogenous factors. This is an image of the chromosome 19, and this is where the tissue kallikreins are located.
There is homogeneity amongst the kallikreins. They all have the same start-stop codon positions. In addition, they all have 5 coding exons, and kallikreins 5, 6, 11, and 14 all have 2 additional extra exons. Down here you can see kallikrein has a unique catalytic triad made up of histidine, aspartate, and serine amino acids, and that contributes to the functionality and the fact that they prefer hydrophobic and basic amino acids. They also have right here this 11 amino acid kallikrein loop. Kallikreins 1 through 3 have that, and that aids in specificity as well as glycosylation. And the kallikrein 6, all the kallikreins are usually normally secreted as an inactive enzyme at first. They have this pre-pro sequence that prevents binding. And initially the pre segment is first cleaved off, and then it's moved outside of the cell. And then the pro segment is then cleaved by another kallikrein member of the family, and it's cleaved into the mature and active kallikrein.
The only differences that we see in the kallikrein family are in the untranslated regions, which this next diagram will exemplify. So all the purple or blue demonstrates shared amino acid sequences between all of the tissue kallikreins and the plasmid kallikreins. So the darker the blue, the more shared the sequence is, and the lighter blue is the less shared. At least one other kallikrein has the shared amino acid. Yellow demonstrates the conservation. So the higher the bar, the more conserved it is, which is also seen by the number of blue in the columns. Now we're going to talk about a little bit of background information. Kallikrein 6 is encoded by the KLK6 gene. There are 3 different types of isoforms produced by alternate splicing, and it's identified in 6 different mammalian species, including cats, dogs, cows, pigs, elephants, and horses, as well as the human species. And based on the location of kallikrein 6, it can participate in different roles.
Going back with the isoforms, there's 3 of them. The traditional isoform is isoform A, and that's found in the Alzheimer's disease patients, their brain tissue. There's variant number 2, which has exon number 3 clipped out, and then there's variant number 3, which has exon 4 cleaved.
This is just a diagram of the 3-dimensional structure of the protein of our enzyme. You can see that the arrows are different beta sheets, and the helices are demonstrated by different helices. And you can see this autolysis loop.
Kallikrein 6 is made up of 244 amino acids, and again, like we said, it's originally created as an inactive enzyme. Right there is the primary sequence. It's just the single 1-letter code amino acids. Right there, the graph to the side is from Uniprot, and it is basically a map of the secondary structure, which includes 5 alpha helices, 15 beta sheets, as well as 4 turns. The secondary structure consists of 12 cysteine amino acids and 6 disulfide bonds. It also lacks the kallikrein loop, which lacks kallikrein loop between beta strands 7 and 6. And it also is a single domain enzyme.
Now we're going to talk about the expression of kallikrein 6 in Homo sapiens. So we see it expressed in a lot of different types of tissues and various fluids. It's seen in high expression in the milk of lactating women, cerebrospinal fluid, and various other serums. It's also seen in glands like the salivary gland, the placenta of babies, the fallopian tube, breast, pituitary, and the kidneys, but more specifically in the nervous tissue. In gray matter neurons, it's seen in high expression, and it's colocalized with Lewy bodies and glial cytoplasmic inclusions. We also see that it's overexpressed in some tumors, but not all, and the expression levels change based on what it's participating in.
Here, this is just where in each individual cell where kallikrein is located. It's located in the cytoplasm, the mitochondria, the nervous system, and then these two pictures on the side, this is showing that kallikrein 6 is mainly located in glial bodies, and then over here is showing that it's mainly located in the exterior processes of the oligodendrocytes.
So now we're going to talk about how it affects the human body.
In some instances, kallikrein is severely underexpressed, and that can lead to buildup of plaques, like the tau plaques in Alzheimer's disease. Because the kallikrein is underexpressed, it's not able to clear out and break down the buildup of substances. And then in other instances, it's overexpressed, like patients with multiple sclerosis, as well as Alzheimer's disease, and because it's overexpressed, it's sort of breaking down all the microtubules on the oligodendrocytes, and it's making it more distance between the synapses. They're not able to connect, and they're not able to make connections, and you're getting loss of sensory function and memory.
Yeah. So Kallikrein 6 is relatively new, so we associate it with a lot of different things based on its expression levels, but we don't really understand how it affects them. We know that it cleaves various extracellular matrix proteins, and that's how it affects metastasis. But we also know that as Kallikrein 6 increases, it also affects the proliferation rates and inflammatory processes within cells, and that's how it affects cancers. Now we're going to discuss our primary article, which is called The Critical Role of PAR1 in Kallikrein 6 Mediated Oligodendropathy. In this article, they were testing the hypothesis that Kallikrein 6 could regulate the oligodendrocyte pathophysiology and biology by activating its various PAR receptors. It was published in GLIA.
So we already knew that kallikrein was severely overexpressed it was like the most, out of all the kallikreins, had the highest concentration in the CNS. And it was especially overexpressed in patients who have had injury in the CNS, like spinal cord injuries, Parkinson's, Alzheimer's. And this article is discussing that kallikrein 6 regulates and activates these PAR receptors, which what it does, it cleaves this tethered ligand, and it promotes dimerization of the PAR receptors, and then the dimerization is what allows the signaling and allows the binding of the protein of the enzyme, and it activates these PAR neurons, and it also contributes to oligodendroglia pathophysiology. So what it does is it reduces the differentiation of the oligodendrocytes, and it also breaks down their processes, and again makes the synapses, it makes it harder for the neurons to synapse.
Yeah, and within the article, they wanted to compare it to something they already knew that activated PAR1 and PAR2, and that's what these little triangles represent. It's thrombin. It's a naturally occurring substrate for the protein receptor. So naturally, it would bind to the receptor and cause it to activate by dimerization, like Amber said, and trigger neurotoxicity. But Kallikrein 6 can intervene without thrombin being necessary and activating the PAR1s, making them think that they've been signaled to induce neurotoxicity, but they haven't. And that's what this diagram further explains. It just shows that exactly where Kallikrein 6 will cleave at that serine residue on the tethered ligand, which just refers to a ligand that's part of the protein itself. And this is a diagram of PAR2. However, PAR2 and PAR1 are basically homologous structures, so it's very similar in the activation processes.
So it also stimulates this ERK1/2 pathway, and that when that's overexpressed, it leads to sort of like a disruption of the myelin proteins.
This experiment used various different methods because they wanted to test multiple aspects of Kallikrein 6 activity. So they used, in order to do that, they wanted to harvest a large number of cells. So they used 8 to 10 week old mice of a certain strain, and they crossed them based on their presence or absence of the PAR1 receptor protein, and they created an offspring generation that they used for their experiments. They anesthetized their mice after 1 day of living after birth, and they used 5 millimeter segments of the spinal cord and grew those cultures on DMEM media. And then they also took the oligodendrocyte progenitor cells, which are stem cells, and grew them on neurobasal A media.
infected them retrovirally with this T-new oncogene, and then they stained and incubated the cells, and then they were able to like fluorescently view and see which areas of these oligodendrocytes had high expression of the KLK6.
Yeah, they use that with pairing antibodies from other species like the rat and goat. And they used Western blot to quantify their data. So this is the first part of their results. They wanted to test exactly where it was located. So you can see in that top left green picture that KLK6 is greatly localized within the cell bodies in the cytoplasm of oligodendrocytes. And then you can see that they tagged PAR1 in these next two. And O4 just refers to a type of oligodendrocyte that they used. And you can see that it's widespread throughout the entire protein. And then they also noticed, which was something interesting, was that PAR2 was 4 times less expressed in the cells, and it was only localized to the cell bodies.
The next feature of the experiment, the study, was they were looking at basically oligodendropathy. So in this chart, in this picture, there's the two controls. So they have two species of mice. They have the wild type mice, which have the PAR1 gene, and then they have mice that don't have the PAR1 gene. And for both the control, this was without the addition of KLK6. They both had great expression of the oligodendrocytes. And then down here, when they added the KLK6 to the mice, the cells that had KLK6, they noticed that there was a significant decrease in concentration of these oligodendrocyte cells, and these processes are actually becoming a lot shorter. So what this whole interaction does is it shortens and depolymerizes and breaks down those microtubules and microfilaments that make up the oligodendrocytes. And again, it makes it harder for them to synapse, whereas this one, it leaves when it's affecting the PAR cells when it's affecting the cells that don't have PAR, they are unchanged because PAR is necessary for the oligodendropathy.
The third part, they wanted to see how it affected the morphology of the oligodendrocytes. So you can see on the top left in figure A, these 4 boxes, there's the different types of morphologies that are known to exist in the oligodendrocytes. There's simple, that refers to the most basic form. Then there's incomplete, where the processes are developing but they're not fully completed, and that's where complete comes in. All the processes have been produced. And then the most advanced morphology is called the membrane morphology, and it's when the entire cell is surrounded by a membrane, including the processes. And so they wanted to test how Kallikrein 6 affected the types of morphologies produced. And you can see in figure B, the top left, this is the control, so you see a little bit of everything. You see mostly membranous and complete, and you see you don't really see incomplete or simple because all of the oligodendrocytes are differentiated. And then you can see as ATP and Kallikrein 6 are joined in the serum that they were growing the cells in, there's a great decrease in the number of oligodendrocyte processes overall. But they also noticed that as the Kallikrein 6 was exposed to the cells, it completely eliminated the membrane phenotype, as well as 11-fold decrease of the complete phenotype, and it increased the number of simple and incomplete phenotypes expressed. And that's what these diagrams in here quantify.
In the last part, they looked at these primary oligodendrocyte precursor cells. These OPC cells are stem cells that give rise to the mature oligodendrocyte cells. And so they wanted to incubate those OPCs with KLK6 to see what the effect would be for that one. And what they noticed was when they infused the stem cells with KLK6, it completely stunted their growth. Like, they weren't even able to grow at all. It wasn't even depolymerization, it was just no growth of the oligodendrocytes. And again, it's 60% fewer processes compared to the controls.
Yeah, and they wanted to compare it, like we said earlier, to thrombin. And then they threw in another kallikrein, which is kind of expressed in the central nervous system, and they saw that Kallikrein 6 defeated them by far.
And it didn't even affect, they also exposed it to Kallikrein 1, which is part of the family, and it didn't do anything.
So in our conclusion, or the conclusion of the article and our overall presentation, is that Kallikrein 6 is a neurotoxic enzyme that causes the destruction of oligodendrocytes by using PAR in the nervous system. And it does this by activating the ERK1 and 2 pathways, which leads to the retraction of processes by breaking down myelin, as Amber said. And then it also reduces the number of differentiated oligodendrocytes and inhibits the outgrowth of progenitor stem cells. These are our sources. Thank you.